So my name is Josefina Nilsson, and I'm the CEO of QuTEM, and you have to bear with me. I came in very late last night from Sweden, but let's try this out. So I will give you a brief introduction to QuTEM and what we do for our clients. So, as I said, we are located in Stockholm, Sweden, and we are a CRO focusing on electron microscopy and image analysis of different kind of nanoparticles. And these are our three cornerstones. Of course, our experts, our personnel, which is not only experts in what we are doing in electron microscopy and image analysis, but also in the particles that our clients are working with. And then we have the proprietary software, which we have developed in-house, which we are using throughout the whole process. We are acquiring the data with it, we store the data, we do the analysis, reporting, and so on. And this analysis tool is Part 11 compliant. And all this is wrapped into our GMP certificate. And this GMP certificate is, of course, important for us, but also for our clients, because that means that we can work with our clients from early R&D through clinical phase, and also do batch release when they have a product on the market. Most of our clients, I would say 50-50, 50% in Europe and 50% here in North America. So we are working with, or our clients are working with, a lot of different particles. So for cell and gene therapy, mainly it's AVs, of course. Is it me that is sounding? No? It's just peeping. So lentiviral vectors, lipid nanoparticles, adenoviruses, and so on. And depending on which particle you're working with, we are measuring different critical attributes for that particular particle. And this is how it goes. Our clients send a very small volume to us in Stockholm, approximately 20 to 25 microliter. We do the sample preparation in our BSL-2 laboratory, perform the imaging there as well, do the image analysis with the Gridsee analysis tool, and report that together with all the acquired images to our clients within 10 business days. We can do this faster also. Sometimes that is needed. We have turned around the project in 24 hours when it's really, really needed. So even though I know that a lot of people are working with different particles, I will today focusing on the AAV vector. What kind of quality attributes do we measure for our clients there? And you know this much better than me. You need to keep track of the full empty intermediates and sometimes also overpacked AV particles that you have in your sample. I mean, of course, you would like to have the full capsid that then give you the functional protein that you would like to have. But very often, or I would say almost every time, you will also see empty particles, broken particles, partial filled, and also overfilled particles. And we are using cryo-electron microscopy and image analysis to, let's say, analyze this. And what we do analyze is the internal density of each and every capsid. And a typical readout from that can look like this. There could be multiple peaks, much more than 3, but in this example we have a small empty peak, a quite large peak, 86% of something that have packed something in the capsid, but also a smaller peak of 11%, which have packed something even heavier. So which is your peak of interest? Where will you find your product? It could look like this, of course, and not so fun, that the majority, the 86%, is then partially filled, and your product is the small peak of 11%. But it can also look like this, of course, where the majority is your product, but then also means that you have 11% of overfilled capsids. So basically, these relative numbers are not really sufficient. So our clients have asked us a long time, can you also then give us the genome length of each and every peak? And yes, we can, because each genome length actually produces a very distinct density signal depending on the genome length. So based on a reference curve that we have built, analyzing a lot of different AV serotypes with very well characterized genome length, we have been able to build a reference curve. And with this reference curve, we can then analyze, let's say, AV particles with, for us, unknown genome length. So going back to the example that we had before, now we have the 3 peaks again. Yes, we have a small peak of empty particles. We have the larger peak that have packed 3.8 kilobases, and the small peak that have packed 4.1 kilobases. And since our clients know the genome length that they expect, this one was quite successful. So the genome interest is actually the big peak, 86%, and then we have a small peak of overfilled particles. And these 2 methods, we have performed a platform validation of both. And as you can see, based on the numbers, it's very, very stable and robust. So when it comes to the vector core, determining the size of each and every population, you can see in the repeatability and intermediate precision that the standard deviation is actually lower than 1%. And when it comes to determining the genome length, the same is less than 150 bases. And it's very sensitive. That means that we only need to analyze or find 50 particles that have packed exactly the same genome length for us to see a signal. However, we are also doing product-specific validations for a lot of our clients right now. We have also done a comparability study together with our collaborator ViralGen in Spain, which have just recently been published. And there that we could see that our method, QuTEM method, together with AUC and mass photometry and HPLC, actually gives very stable results. However, sometimes the QuTEM method, the cryo-TEM method, gives you a higher number of empty particles. And we started to look into that. And we think that that is actually due to particle aggregation, which is then another quality attribute that you need to take care of. So we have a statistical method where we can measure the theoretical distance between each and every particle, depending on the number of particles in a specific area, and then determining the individual particles and also the ones that have aggregated or clustered. And what we found out is that TEM is particularly powerful when it comes to the small aggregates, let's say particles around 3 to 7, 10 particles. And of course, we can measure the amount of aggregated particles, and we can also measure the size or the area of these aggregates. So we can then give you the number or percentage of individual particles and the ones that have clustered or aggregated. And since we can then determine or detect the particles also in the clusters, we can then also add on the tool of determining empty, full, and intermediates of this. And going back to what we found out in the comparability study, we could then see that the number of empty capsids are higher in the aggregates or clusters compared to the ones that are seen as single entities. And that's, I wouldn't say that it's not that surprising. I mean, when you have capsids that are not as stable, which we know that the empty ones aren't, they have a tendency to break and then also cluster. So we will measure these ones, but for instance, in the AUC, if you have clusters or aggregates, they will just pass through the analysis and not end up in that single band of empty particles. So I hope that I've been able to very, very fast show you that you can get a lot of information out of cryo-electron microscopy in one single run. I mean, from one image, one data set, you can start to look into the integrity of the particles. Are they intact? Are they broken? You can also look into the morphology, which we haven't even talked about here. Do you find these doublets or triplet particles? You can measure the vector content. What kind of populations do you have in your product? And you can also then measure the genome length of each and every subpopulation. And you can also look into the aggregation state of that particles or that sample in different ways. And you can have this in 10 business days if you only send 25 microliter of your precious samples to us in Stockholm. But I think and I know that we very, very soon, you don't need to send all the samples to us in Stockholm. You can actually send, especially your R&D samples, to different facilities here in US. Because right now we are starting a collaboration with core facilities in Boston, New York, and Philadelphia. They will do the sample preparation and image analysis, or sample preparation and imaging according to our methods. And then they will send the images to us in Stockholm, and we will do the analysis and reporting. And as I said, it's only for the R&D grade samples, but it will be very easy to do tech transfer for, let's say, GMP samples when you are there, because then you will have to send those to Stockholm. And this will be super easy since these core facilities have the same instruments as we have, and they are using our methods. So, by this, thank you very much for your time. I'm happy to take any questions if you have any, or you can pass by our table, which is very close to the partnering area in the MacArthur room. Okay, thank you very much.